This is, I think, in the last video of this lecture, we will talk about plant mitochondrial electron transport, or we call it electron transport chain of the plants. This picture is quite difficult to get into, but I will simplify it for you guys. So first, let me bring up my pointer. All right. So this picture combine everything together, and from glycolysis to citric acid cycle, including the pentose phosphate pathway. Apart from making ATP, those three pathways give you the reducing power NADH, FADH2, and NADPH from pentose phosphate pathway. Those three reducing power can release their electron through the electron transport chain that occur in the in the inner membrane of the mitochondria, and that will create the chain differences of concentration of the proton between intermembrane space and matrix, and those drive the synthesis of ATP. So let's come back to the structure of the plant mitochondria again. So mitochondria composing of two membrane system, outer and inner membrane, and you get at least two spaces, the matrix and the intermembrane space right here. So here is the inner membrane. This one is the outer membrane. Intermembrane space is somewhere in the middle of the two membranes, and matrix is the innermost layer. So in plant mitochondrial electron transport, which is similar, I can say it's the same with other organisms, we will have four major protein components. We call it complexes because each contain different molecules, many molecules of proteins working together in the form of fourth structure protein, quaternary structure to be clear. Complex one is NADH dehydrogenase. It can catalyze the losing losing electron reaction of NADH to become NAD+. Complex 2 is the succinate dehydrogenase. It can catalyze the reaction of transferring electrons from succinate to fumarate to produce from NAD FADH2 to FAD. Actually, I think I switched the order. Here you will start with FAD and you end up with FADH2 carrying electron, and electron from FADH2 will enter the complex 2, and those electrons can be both from complex 1 and complex 2 can give electrons to plastoquinone, which is an oxidized form, and once the plastoquinone receives electrons, it will become reduced form or plastoquinol. We call it UQH. If you may recall from many pharmaceutical products, it will advertise that it will have coenzyme Q or Q10. Is Q10 or coenzyme Q is actually the other name of ubiquinone, which is the antioxidant. It will receive electron and is involved in electron transport chain of human as well. So complex 1 and 2 transfer electron from NADH and FADH2 to plastoquinone and become plastoquinol, the reduced form. And plastoquinol is then transferred those electrons to complex 3, which is cytochrome BC1 complex. This cytochrome BC1 complex or complex 3 will transfer that electron to cytochrome C. So it will use the ubiquinol as the electron donor, and once it takes electrons from plastoquinol, plastoquinol will become plastoquinone again. That can receive more electron from complex 1, complex 2. Alright, let's come back to cytochrome C. Cytochrome C can receive one electron at a time, and then the reduced cytochrome C will donate electron to complex 4. Complex 4 that carry electron will transfer that electron to the final electron acceptor, which is oxygen. For every 4 molecules of cytochrome C oxidized, 1 molecule of oxygen is reduced and 2 molecules of water will be produced. Alright, this is the major pathway for plant electron transport chain. But you can see that plastoquinone is quite a center of everything. It receives electron from both complex 1 and complex 3. And complex 4, the name of the alternative name is cytochrome C oxidase. It means it will oxidize, it will give electron to oxygen and produce water. So we have an alternative pathway for producing to giving electrons to plastoquinone. We can have NADH or NADPH dehydrogenases from the matrix site or from the intermembrane spaces that can oxidize NADH or NADPH and transfer those electrons to plastoquinone. The same from the matrix site. And apart from complex 4 that can transfer electron to oxygen and produce water, we also have the alternative oxidase that can receive electron and hydrogen from plastoquinol and give that to oxygen and produce water. And by this process, the alternative oxidase, not like the other complexes in the electron transport chain, it will not produce ATP, the process of the free energy released as the electron flow is lost as heat. Alright, as I mentioned earlier, the alternative oxidase cannot produce ATP, but some complexes like complex 1, complex 3, and complex 4 can produce a substrate for making ATP. These 3 complexes, by transferring electron, it will have some energy, and that energy can pump proton from matrix into intermembrane space, creating the differences in the concentration of proton gradients. We call that as the proton motive force that will drive the ATP synthesis. Since you have more proton concentration in the intermembrane space, this proton will enter the ATP synthase and go back to the matrix. By transferring proton from intermembrane space to the matrix, this part of ATP synthase will rotate, and by the process of rotation, it will create energy to combine phosphate and ADP or adenine diphosphate and produce adenine triphosphate or ATP as the cellular energy. And this is the whole process that I try to simplify for you guys in this lecture. If you look at the capacity to lose electron, here the higher the electron volt, it has more capacity to lose electron, and the lower it has more capacity to receive electron. We call it oxidizing power. This one has stronger oxidizing power, and the upper one has stronger reducing power. It can reduce other components better. So NADH has the strongest reducing power, give electron to complex 1. Complex 1 will lose electron to plastoquinone, and succinate or NAFADH2 will give 2 electron to complex 2. Complex 2 will transfer those electrons to plastoquinone. Plastoquinone transfer 2 electron to complex 3, which has lower reducing power. Complex 3 will lose electron to cytochrome C. Actually, cytochrome C can travel inside the intermembrane space, but complex 1, 2, 3, and 4, they fix into the membrane, they cannot go out. But cytochrome C, they are already out, they can move along. So this cytochrome C will move the electron from complex 3 to complex 4, and complex 4 can transfer the electron to the last electron acceptor, which is the oxygen. And oxygen will combine with hydrogen or proton with electron and produce water, which is the lowest in reducing power in this scheme. So right here you need to compare the electron transport chain in mitochondria and chloroplast. So let's start with where everything is. In the mitochondria, the membrane that housing the electron transport chain is inner membrane. In chloroplast, it occurs in thylakoid membrane. For the space that has more proton, it is intermembrane space in mitochondria, which is right here outside of between the outer and inner membrane. For chloroplast, it's actually the innermost space, which is the lumen inside the thylakoid membrane. And the ATP will produce in the lower concentration of proton, which is in matrix, the innermost space in mitochondria, and in the stroma right here of the chloroplast. Here is the comparison between where the ATP is produced between mitochondria and chloroplast, but I expected you to know more. You need to compare each component, like complex 1, complex 2, 3, 4, cytochrome C, ubiquinone, with from the mitochondrial electron transport chain to the chloroplast electron transport chain, starting from photosystem 2 to plastoquinone to photosystem 1 to NADP plus and to NADPH. You need to compare NADPH and water to the oxygen and NADH or of mitochondria as well. And if you look at the outcome of the oxidation of sucrose, you start with 1 sucrose. In the process of glycolysis, you get 4 pyruvate and 4 ATP, 4 NADH, and for here you have 4 ATP as the substrate level phosphorylation from glycolysis. For citric acid cycle, you start from 4 pyruvate. You get 12 carbon dioxide, 4 ATP, 16 NADH, and 4 FADH2. Here you have 4 ATP from citric acid cycle and 4 from glycolysis. These are substrate level phosphorylation. It is the direct phosphorylation of ADP using energy released from other reactions. And from NADH and FADH2, you get around 60 to 64 ATP in the oxidative phosphorylation. It is the synthesis of ATP from oxidation of NADH and FADH2. Alright, this is all about electron transport in the plant's mitochondria. Here I have some questions for you guys from lecture 3 photosynthesis and lecture 4 respiration for you guys to think about to prepare for exams. So the first one is, how does the plant mediate electron transport in the chloroplast? Why is magnesium is important for chlorophyll? Why do plants carry many types of photosynthetic pigments? Differences between linear and cyclic electron flow in chloroplasts. Why Rubisco is the most important protein on Earth. Similarity and differences between C4 and CAM photosynthesis. Differences between plant and animal respiration. Similarity and differences between photosynthesis and respiration. What are the last electron acceptor molecules in the light reaction of photosynthesis compared to mitochondrial electron transport chain? The same question. What are the electron donor in the photosynthesis and mitochondrial electron transport chain? How all the processes learned from this lecture works? Where all the processes learned in this lecture take place? What are the part of the cells? Functions of ATP and reducing powers. Importance of cyclic electron flow and oxidative pentose phosphate pathway. I can tell you that oxidative pentose phosphate pathway produce NADPH but do not produce ATP. For cyclic electron flow, you produce NADPH as well, but do not. No, it's the opposite. Cyclic electron flow produce only ATP but not NADPH. See, I can miss the point. So here I give you an answer already because it's quite difficult for you guys to figure out. So I decided to give you an answer. The next question, what is the mechanism driven ATP synthesis by ATP synthase? This is very easy. I think I mentioned it a lot. And the next one, what is the difference between substrate level ATP synthesis and oxidative phosphorylation? I have just mentioned it in the previous slide. And finally, why do we need to study these difficult processes of plant and living organisms? This is quite an open question. Think about it. It's quite a bonus, I think. And here are the references that you can go after, can read, and you can gain more understanding from them. Otherwise, there's a lot of YouTube's video that give much more better explanation or animations for you guys to study. But remember, respiration in plants is not quite identical with other organisms. At least it starts from sucrose and it will end up with pyruvate and malate, not only pyruvate. Alright, I think that's all I have for this lecture. See you guys next week. Have a good one.